Quickly, we'll go through hypersensitivity reactions. Four types of hypersensitivity reactions are there, and these hypersensitivity reactions are basically inappropriate immune response. It's not that the response is always aggravated, but mostly the immune response is inappropriate. And since the immune response is inappropriate, it may result in undesirable outcomes in terms of disease or tissue destruction. So we'll talk today first about type 1 or IgE mediated or immediate type of hypersensitivity. So this is first type 1 hypersensitivity. So here what happens, the antigen in type 1 hypersensitivity is referred to as allergen. Why it is referred to as allergen? Because it results in allergy. So type 1 hypersensitivity is also called as allergy. So this is an allergen, and suppose an allergen enters the body and B cells then encounter that allergen. B cell would recognize that allergen through their B cell receptor, and then this allergen would be taken inside by the B cell, because B cell can act as antigen presenting cells also. And this allergen peptides then would be displayed on the MSC2 molecule, which is present on antigen presenting cell like B cell. So this antigenic peptide will be recognized by the TCR receptor, T cell receptor, and the T cells that will recognize it will have CD4 receptor on them. So CD4 recognizes the MSC2, and the TCR will recognize the antigenic peptide. And because of this T helper cell would get activated, and it will differentiate into memory cells and plasma cells. Plasma cell will produce IgE. So normally what plasma cells should produce on primary exposure? They should produce IgM. But in this case, instead of IgM, IgE is produced, and this IgE is specific to this allergen here. So once this IgE is produced, this IgE can go and bind on mast cells or on basophils onto Fc epsilon receptor. Fc epsilon receptors are the receptors that can bind IgE. So this IgE goes into the circulation and it can bind to the mast cells or the basophils. And then once it binds to the basophil or the mast cell, these cells are called as sensitized mast cells or sensitized basophils. Now think of a situation where this individual is second time exposed to the allergen. So now there is an entry of the allergen again, same allergen, and now these cells are already sensitized. So this allergen will go and it will bind with the IgE, which is already bound to the Fc epsilon receptor. As the allergen binds to these IgE molecule, because of their cross-linking, the degranulation of the mast cells and the basophils happens, which release number of vasoactive amines like bradykinins, the cytokinin, histamines, and all such molecules, leukotrienes, they will be produced. And these vasoactive amines, they'll act on smooth muscles, causing their contraction. They can act on small blood vessels, causing the vasodilation, and they'll act on mucous glands, causing them to become hypersecretory. Even blood platelet clotting would happen, and such as eosinophil will also be getting activated. They'll release eosinophil chemotactic factors, neutrophil chemotactic factors, and all these outcomes will result in certain kind of disease. For example, atopy, yes, then you have asthma, you have hay fever, rhinitis, allergic rhinitis. So these are all outcomes of these chemical mediators which are released, and they act on different blood vessels and tissues. Depending upon the site, they will cause the different types of disease conditions. And these are local. This is localized anaphylaxis, you may say. And when there is anaphylaxis, you can have systemic anaphylaxis. In systemic anaphylaxis, for example, in case of venom from bee sting, then the systemic anaphylaxis reaction, that is more like a shock, can happen, and this could be fatal as well. So this is how type 1 hypersensitivity is manifested. It's the allergen and the production of the IgE, and these IgE then binding to their receptors on mast cells, and when the second time allergen enters, this binds to the IgE. Those are already produced against the allergen, and they are bound to the mast cells, and as the allergen binds, there is degranulation, and because of this, many chemical mediators are released, and they act on different cells, and those the outcome of those actions are production of the disease such as hay fever, asthma, rhinitis, and even the systemic anaphylactic shock. Now talk about type 2 hypersensitivity. Type 2 is also called as IgG mediated cytotoxic hypersensitivity. Remember that IgG is the predominant antibody that is responsible for type 2 hypersensitivity, although in some cases IgM is also involved, but in most of the times it is the IgG that is responsible for this cytotoxic hypersensitivity or the type 2 hypersensitivity. So one such example of this is erythroblastosis fetalis. Okay, in erythroblastosis fetalis, Rh negative mother, when is bearing a child which is Rh positive, that means the RBCs of that child carries Rh antigen, whereas the mother RBCs are devoid of Rh antigen. So normally during the pregnancy, this the fetal circulation is separated from the maternal circulation through these trophoblast here. But at the time of delivery, some of the blood from the umbilical cord enters into the mother's circulation, and when these RBCs are in the mother's circulation, the fetal RBCs are in the mother's circulation, then these are picked up by the B cell, which are specific to the Rh antigen. So these B cell, those are specific to the Rh antigens, then can secrete, then they can change themselves into the plasma cell and start secreting IgM. This IgM would be eventually cleared from the mother circulation. Apart from plasma cell, memory cells are also formed, and as you know that memory cells, they have longer lifespan, so they are going to be inside the mother circulation for long period of time. So what happens when there is a second pregnancy, and similar situation is there when the fetus, which is Rh positive. And why fetus is Rh positive? Because the father is Rh positive and mother is Rh negative. So fetus can be Rh positive. And during the second pregnancy, some of the blood may enter into the mother circulation. Here also some blood enters, but the amount of the blood entering into the mother circulation is not enough so as to induce immune response. But during the delivery, more blood enters, and then the immune response gets activated. Whereas in the second pregnancy, even small amount of blood entering from the fetus into the mother, already memory B cells are there, and you know that memory B cells, they will be easily activated by the even the small amount of the antigen. So even the small Rh positive RBCs, small number of them, they'll make memory cells to clonally expand and become plasma cells. These plasma cells would now secrete IgG. On the primary response, IgM was secreted. On the secondary response, IgG is secreted. So IgG mediated. So IgG then can enter through the placenta into the blood of the fetus, and these antibodies then they can bind to the RBCs onto the Rh antigen of the fetus, and they can activate complement, and complement mediated lysis of RBCs can ensue. And depending upon how much antigen antibody complexes are formed, the destruction of RBCs can happen. This may be life threatening, or it may be just causing mild anemia or jaundice. But this all can be prevented in the second pregnancy if we know that this situation is there. Immediately after the first pregnancy, first delivery, antibodies against the Rh antigen, they can be injected into the mother, and these antibodies, they can go and bind with the Rh antigen uh, in the they can go, and so they'll be in the mother, and whenever that blood will enter into the mother circulation from the fetus, these antibodies will readily go and bind with the Rh antigen. So this Rh antigen will not be available to activate the memory cells, and this thing here will be not happening. So the erythroblastosis fetalis can be prevented by injection of these antibodies against the Rh antigen in the second pregnancy. So beside this, there are other examples like an hemolytic disease of newborns. For example, hemolytic disease of newborns. For example, in foals, pigs, and other animals, this can happen. So what happens in foals, for example, the antibodies, they cannot cross the placenta, but these iso antibodies, they are present in the colostrum, and when colostrum, the foals drink this colostrum, they can then enter the circulation and can cause this lysis of the RBCs also. So it's pretty much same way, and the mechanism would be pretty much the same way as it is in case of this erythroblastosis fetalis. And then you can think of a situation where there is blood transfusion. So you can have incompatible by mistake. There is if incompatible blood transfusion. Suppose the incompatibility is between the major antigen ABO type incompatibility. Then the IgM, for example, individual having blood group A, if injected with blood group B, then that individual with blood group A will have antibodies against the blood group B, and these antibodies can bind with the RBCs, and they can activate the complement. And in ABO type incompatibility, it is usually the IgM isohemagglutinin that goes and bind with the RBCs. Okay. Whereas if minor blood group incompatibilities are there, minor blood groups in humans are like Duffy is there, Kid, Cal. So these are minor blood groups, and usually they do not induce any transfusion reaction if you're injecting like if your transfusion is being done only 1-2 times. But in individuals, which require multiple transfusions on multiple occasions. These the antibodies to these antigens, incompatibility of the antigen, for example IgG antibody then can bind with the incompatible blood group, and there will be lysis of the RBCs, again through complement mediated lysis or through antibody dependent cell cytotoxicity. So the lysis here, the cytotoxic effect, the lysis happen because of complement or through antibody dependent cell cytotoxicity. So once the complex is formed, like here, the complement can come and it can bind and it can activate complement pathway. And you know that in complement activation pathway, there is formation of membrane attack complex, which causes pores in the membrane of the cell, leading to its destruction. So that's why it is cytotoxic hypersensitivity. Same way, the cells can be destroyed by antibody dependent cell cytotoxicity. It means that this cell cytotoxicity is also dependent on antibody. So when the antibody is bound to this any cell through the antigen, right, then these antibody molecules, they can attach to the Fc receptor on certain cells, for example NK cell. So this NK cell can then induce this cytotoxicity of this affected cell. So type 2 hypersensitivity is IgG mediated, except in some cases, as in a case of ABO incompatibility, if the transfusion is happening for the first time, then it is IgM, but mostly it is IgG on almost all the occasions. So these are some of the examples that I gave you. Hemolytic disease of newborn. Then there is another example, drug-induced hemolytic anemia. Drug-induced hemolytic anemia. So certain drugs like penicillin and cephalosporins, they can bind with the proteins on the RBCs, and once they are bound with the protein on the RBCs, and they act like hapten carrier molecule. Penicillin on its own will not activate B cells to produce antibodies, but when penicillin is bound with the protein on the RBCs, this protein penicillin complex can activate, they can act like an immunogen, and they can activate the B cells, and the antibodies would be generated, and these antibodies then can bind with the drug protein complex, and in turn activating the complement and causing the lysis of RBCs. That's why, if you remember, that doctors always ask the patients that are you allergic to this penicillin or a particular drug, because when they are about to inject that particular antibiotic in the individual, they should know whether that individual is allergic to that or not, because certain antibiotics, they can go and bind with the RBC proteins and can cause these reactions, again causing lysis of the RBCs, leading to anemia and leading to the production of hemoglobin, and also leading to hemoglobinuria. So this is type 2 hypersensitivity. We now move to type 3 hypersensitivity, which is called as immune complex mediated hypersensitivity, or type 3. Again, very briefly, if you remember, the here see antigen and antibody, when they bind with each other, they combine with each other, this is called as complex, immune complex. Now the purpose of the immune complex is that it can be easily phagocytosed by the phagocytes, and the antigen then can be cleared from the circulation, because the complexes, they are larger in size and they are easy to phagocytose, and this is one of the way to remove the antigens or to get rid of the antigens from the system. What in some cases, these complexes here, they basically uh, are formed in huge numbers, and when they are formed in huge numbers, the phagocytes cannot remove them from the circulation or from the site of their deposition. Hence, when they are not being removed, that leads to the problem. So, for example, look at this diagram here. First is immune complexes are deposited in the wall of blood vessel. So, this is one of the example. This is a blood vessel, and these immune complexes are getting deposited on the basement membrane of this blood vessel. What happens? Because of the presence of this immune complex, this will activate complement. Okay, as you know that complement activation in classical pathways happen when antibody and antigen complex is formed. So there will be activation of the complement, and once there is an activation of the complement, this leads to the inflammatory cells attraction to the site. For example, neutrophils would be attracted. Why? Because complement split products C3A, C4A, C5A, these are, they act as anaphylotoxins, and anaphylotoxins are the molecules that are responsible for attracting the neutrophils to the site of immune complexes, or to this towards the site of inflammation. So when these neutrophils, they come to the site, look here, they try to phagocytose these immune complexes. But since the immune complexes are attached to the basement membrane, they are like large immune complexes, they are attached to the basement membrane. In an attempt to phagocytose these immune complexes, the neutrophil fails and they start to release enzymatic granules, and these granules then damage the nearby tissue, producing a disease or tissue destruction. So wherever these immune complexes are deposited, for example, if they are deposited on blood vessel walls, they lead to vasculitis, rashes, erythema, edema. They can be deposited in synovial membranes, synovial membranes of joints, glomerular basement membrane of kidneys, choroid plexus in the brain. So think of a situation if the immune complex is generated inside the blood. So it will travel through the blood and can settle down in synovial membrane, in the basement membrane, or it can even pass through the blood-brain barrier in choroid plexus, blood vessel walls, and depending upon where these immune complexes are generated, the neutrophils would be attracted to the site, and they can cause the damage. So you can think of arthritis, right? For example, in case of synovial membranes. So this is how wherever the immune complexes will be deposited, it can also lead to the autoimmune diseases. For example, systemic lupus erythematosus. In systemic lupus erythematosus, the antibodies are generated against the DNA. So DNA, anti-DNA antibody complexes can deposit in different places. They can deposit in kidneys, leading to kidney failure, they can deposit, these complexes can deposit in synovial membrane, leading to rheumatoid arthritis or arthritis. This is also, so many autoimmune diseases are because of the antigen immune complex mediated hypersensitivity. Okay, so you have to remember that type 3 hypersensitivity happens because of the deposition of immune complexes in various organs, and these immune complexes are not removed. Neutrophils try to remove them, they try to phagocytose them, but they fail. And since they release a lot of granules, these granules are rich in proteases and other enzymes, and they can damage the nearby tissue and hence lead to different types of diseases. So finally, we talk about type 4 hypersensitivity. Type 4 hypersensitivity is also known as delayed type of hypersensitivity. Delayed type of hypersensitivity, or also cell mediated hypersensitivity. Here there is no role for antibodies, no role of antibodies, only cells. And cells would be usually macrophages and T helper cell, T helper 1 usually, right? Macrophages can also result in epithelioid cells, giant cells, okay, fused macrophages. So these are the cells that are participating in this cell mediated type of hypersensitivity. And this delayed type of hypersensitivity, DTH, was earlier known as tuberculin hypersensitivity. Tuberculin hypersensitivity. Why? Because Robert Koch, I think 1890, need to check, when he observed that when he injected tuberculin is a purified protein derivative of mycobacterium. He did not use purified protein derivative, but he used the filtrate of mycobacterium tuberculosis to inject intradermally the persons who were suffering from TB. So the persons who were suffering from TB, when he injected intradermally this filtrate of mycobacterium tuberculosis, he found that at the site of injection there was inflammation, there was edema, there was erythema, nodule-like tubercle formation happened there. And so he called this reaction as tuberculin reaction. But later on, the name was changed to delayed type of hypersensitivity because other antigens, when injected into certain individuals who were suffering, who were already sensitized to that antigen, the similar kind of reaction was seen in those individuals as well. And hence the name was changed to delayed type of hypersensitivity. Why we call it as delayed type of hypersensitivity? It is for obviously one reason, that the time it takes to display the reaction, to form the reaction is around 72 hours, or usually 48 to 72 hours. So it's the long time that it is required for the cells to come and to effect the reaction. So because of this is called as delayed type of hypersensitivity, the time it takes to cause the destruction of the local tissue. Okay, so in type 4 hypersensitivity, if we look at the pathogenesis of type 4 hypersensitivity, we have two phases: sensitization phase and effector phase. So what is sensitization phase? So think, for example, let's talk again about, suppose, mycobacterium. An individual got infected by a mycobacterium bovis. Let's talk about cattle. So once the mycobacterium bovis gets into the cattle, get inside the cattle, then the it will be the bacteria will be uptaken by the antigen presenting cells, for example macrophages, and these macrophages would then present the antigen to the T helper cells. T helper cells would secrete cytokines which would proliferate, cause the differentiation and proliferation of these T helper cells into T helper 1 cells. These T helper 1 cells are also called as DTH cell or delayed type hypersensitivity cells, but obviously they are the subtype of Th1 cells. So it is usually the Th1 cells. The DTH cell as such, we don't have any separate functional population, but they are referred to as because they are participating in the delayed type of hypersensitivity. So once the antigen enters in the body, the T helper cells pick up that antigen. It recognizes that antigen in association with the MHC, and then they become sensitized. In a way, now the cells, T helper 1 cells, are sensitized. So this takes around maybe 2 weeks for the sensitization phase to happen. Now the animal is said to be sensitized. Okay, so after suppose 2-3 weeks, if second time the antigen enters, for example, now if we want to do tuberculin testing, or when the second time the antigen would enter that individual, then you will have already these sensitized Th1 cells will already be there. Okay, and these cells then, once they get second exposure to the antigen second time, these T helper cell Th1 cells would start secreting cytokines like TNF beta or interferon gamma. Other cytokines are here like IL-2, IL-3, IL-8, and these cytokines then they can act on the resting macrophages. Okay, so macrophages which are not activated, even these cytokines, some of them they can attract the monocytes from the circulation into the tissue spaces. So there will be a lot of macrophage would come to the site, and these macrophages would then become activated. The activated macrophages would they'll enlarge in size, they'll display many receptors on their surface, and these receptors then become susceptible to different or sensitive to different types of cytokines. For example, here TNF receptor, if the cell has got more TNF receptor, it becomes more sensitive to TNF. If it has got more MSC2 molecule, it will be able to present the antigen effectively to the T helper cells. So macrophage activation happens, and these macrophages then migrate to the site of the antigen. More monocytes come and they surround the antigen, and when a lot of macrophages are there, definitely they are going to release certain molecules and enzymes and mediators, and that also result in little bit of tissue destruction, and they surround the antigen. So as a result there will be tubercle formation, and if we look at this kind of tubercle, then we'll see that around 5% of these cells are only Th1 cells, antigen specific Th1 cells, only 5%. 95% of the cells in the infiltrate would be non-specific cells and macrophages and maybe the giant cells. So this type 4 hypersensitivity is totally caused by the infiltration of the cells, activated macrophages and T helper 1 cells, T lymphocyte cells. Okay, and it is not always that this type of response is always detrimental to the individual. These are very important. This type of reaction is very important to against the defense against intracellular pathogens like Brucella is also intracellular, or intracellular fungi, even Mycobacterium tuberculosis, Mycobacterium leprae. So these are all the intracellular pathogens against which this kind of reaction happens, and that leads to the capturing of these intracellular pathogens at a particular place does not allow them to infiltrate to various organs in the body, and hence it enables to control the infection. Uh, Type 4 hypersensitivity can also happen because of certain chemicals, certain poisons like contact dermatitis. Contact dermatitis. If you come across, for example, poison ivy, जिसको हम लोग बिच्छू बूटी बोलते हैं, so that kind of reaction is also type 4 hypersensitivity. Okay, even certain individuals are allergic to metal, and for example jewelry, and people wearing jewelry sometimes gets this contact dermatitis, and in that case also there is type 4 type of hypersensitivity. So this is about type 4 hypersensitivity. As I in the beginning I told you that there are 4 types of hypersensitivity, but in some text you would find another type of hypersensitivity, type 5 hypersensitivity, which is also called as stimulatory hypersensitivity. It is not exactly stimulatory, and in some books they refer it as a subtype of type 2 hypersensitivity. Subtype of type 2 hypersensitivity. I'll give you an example of what happens in this hypersensitivity. Is basically the antibodies, they mimic the ligand. So ligand receptor interaction. For example, in case of Graves' disease, which is a disease of thyroid. So what happened? Thyroid stimulating hormone goes and bind with its receptor on thyroid, and hence release the thyroid hormones, release of thyroid hormones. So, but in case of Graves' disease, the antibodies are formed which can mimic TSH, and they can bind with the receptor, thus mimicking TSH and leading to the excessive secretion of the hormones. And as a result, the Graves' disease happens because of hypersecretion of these hormones. In myasthenia gravis, there is improper muscle movement and weakness of muscles is there. So in myasthenia gravis also the antibodies, they go and bind with the acetylcholine receptor and blocks the action of the acetylcholine on the acetylcholine receptor. Hence the muscles, they do not contract, and there is generalized weakness in myasthenia gravis. So it is, in this case it was like stimulatory, whereas in myasthenia gravis it is sort of inhibitory in nature. So these are a bit confusing classification of type 5, but you should know that it is again a kind of inappropriate immune response. Not exactly the immune response, but the antibodies are going and binding to the receptors instead of their actual ligand binding to the receptors and causing these kind of things. So this is all for today. Thank you.